So, hello to all of you. I'm the president of the French Society for Cell and Gene Therapy. And the board and myself are very happy to present a series of webinars on gene editing and gene therapy. We want to give in particular the chance to young researchers, such as PhD students or postdoctoral students. We want to give them the opportunity to present their work here. And the two previous webinars were already presented by Juliette Rosier, who gave a nice introduction on this subject. And the second one was by Laura Ugalde on gene editing and how to cure in this way a rare disease, Fanconi anemia. So I invite you also to go back to those webinars. But today, the current webinar will be presented by Maria Silvia Roman Ascona. She obtained a bachelor degree in biochemistry in Paraguay, where she got to work with phage display technology for bioremediation of heavy metals like cadmium. Then she moved to Barcelona, where she worked on more the field of molecular biotechnology. And she characterized new targets for the treatment of cardiac inflammation, so something completely different. And then she went back to Paraguay and continued working on phage display for bioremediation purposes. And the work she's going to present today is the work she did in Germany for her PhD. And here she worked again on completely something else, which is epigenome editing for CAR T cells. She's working in this lab as a postdoc, where she also did her PhD. So please, Maria, I give you the opportunity to present your work today.
Thank you. Thank you very much, Els, and thank you to the French Society for inviting me to this series of webinars. So as the nice introduction done by Els explained already, I will be talking to you about epigenome editing and its application in CAR T cells. And I thank you all for joining this afternoon. So I would like to start by introducing the epigenome, which literally means above the genome. And from a very conservative definition, we can say that the epigenome is the collection of chemical modifications, also known as epigenetic marks, to the DNA and histone proteins that can regulate the expression of genes from within the genome. So this, as I said, is very conservative, and let's not forget about the recent introduction to non-coding RNAs and also the 3D chromosome folding into the field of epigenetics. But let's say from a more practical way, what the epigenome does is to tell the genome what to do, what to express, where to express it, and when to express it, such as so that we can have from the same genotype several different phenotypes. And these, we as human beings and multicellular organisms are a very good example of what the epigenome can do. So the epigenetic marks, as I said, are chemical modifications onto the DNA and can be classified as DNA methylation or histone post-translational modifications. So the post-translational modifications that the histone can suffer are acetylation, phosphorylation, methylation, and some others. And what this means is that we can have either activating marks or repressive marks. So the activating marks can be deposited onto the histones or the DNA, and are associated with gene expression, such as having a hypomethylated promoter or enhancer region. And the repressive marks that can also be deposited in the histone tails or can also be onto the DNA. For example, the hypermethylation of these promoters or enhancer regions are associated with so with silencing of or repression of the gene expression. Like these epigenetic marks can control the gene expression. But how do they do so? And they do so via epigenetic regulation, which is a language that no one can fully speak yet. But I think we all agree in the fact that there are some key players of the epigenome that are classified as writers of epigenetic marks, which are components of the epigenome that can deposit these epigenetic marks, such as DNA methylation or histone modifications, as I said previously, or erasers that can eliminate such marks. And then there are also readers, which will be interpreting these marks and then conducting a functional outcome. So you can imagine that this is a very dynamic process, as living organisms have to adapt to different environmental conditions. But nevertheless, some epigenetic marks can be inherited during cell divisions. And this has been very important, and nowadays we are acknowledging this more and more to understand the contribution it had to evolution itself. And it allows us to even think of performing what we call targeted epigenome editing. So epigenome editing would be then the addition or the removal of epigenetic marks at a desired locus to specifically modulate the gene transcription without having to modify the underlying DNA sequence. And to do so, we would need an epigenome modifier. An epigenome modifier has to be constituted of at least two domains, one programmable DNA binding domain and one or more epi-effector domains. And I will be describing them with a little more details now. So the DNA binding domain is the part of the epigenome modifier that can be programmable to target the desired sequences in the genome. And these can have different nature. So it would be a recombinant protein, such as zinc finger proteins or transcriptional activator like effectors, or better known as TAIL, which are modular platforms that can be customized to target a specific region of the genome, or can also more recently be relying on the CRISPR-Cas9 RNA-guided DNA targeting, which most of the cases are using an catalytically inactive version of the Cas9 called dead Cas9, and a guide RNA to locate this protein at the desired locus. But where do we target this DNA binding domain? In most of the cases, ideally, we should be targeting them towards gene regulatory regions, such as promoter or enhancers. And the likelihood of the epigenome editing to succeed increases when we know that those areas are susceptible to be regulated by certain epigenetic marks. Now, when we talk about the effector domain, this is the part of the molecule that determines the effect. Either we mostly will want to do either of these options, which are gene activation or gene silencing. This is what we can manipulate with the epigenome editing. And the nature of the effector domain can be related to transcriptional modulation, which elicits gene activation or gene repression. And as a consequence, they end up altering the epigenome. Or it could be a catalytic activity, which will by itself directly deposit or remove a specific epigenetic mark. So they both end up producing the desired change, but they have different ways of achieving this. And just to give you some examples, because there is a plethora of options out there, and if you make a quick search, you will probably find a lot of explorative papers where they report several different effector domains. But for example, as a transcriptional modulation, one of the most common domains is the VP64 for activation, and the catalytically active for activation would be TET1 or P300. So TET1 will act on DNA B methylation, and P300 will act as histone acetylation, or inducing histone acetylation. And if we want to achieve silencing, the most common transcriptional modulation factor will be CRAB, so the cruppel-associated box. And in terms of a catalytic activity that we can take advantage of, we can think of DNA methyltransferase, such as this de novo dimethyl-3A, or also histone deacetylations, or histone deacetylases as HDAC3, which has also been reported. So what are the advantages of doing epigenome editing? And to better explain this, we have to maybe compare this technology to other approaches that are available to modulate the gene expression or gene function. For example, we can think of when we want to delete or abrogate the function of a certain protein, or maybe a certain receptor. In this example here, it's PD-1, widely known in immune checkpoint. We can think of adding a blocking antibody that will interrupt the interaction between the receptor and its ligand, and thus, without changing the gene expression, will abrogate its function. Another, and maybe the most obvious thought you had in these times is gene editing. So inducing gene knockout, for example, by using a nuclease as the famous CRISPR-Cas9 system to directly cause some double-strand break in the genome at the desired locus to induce frame shift mutations that will end up causing the production of a non-functioning protein or an incomplete protein that cannot exert its function. Or we can also think of the RNA interference technology, which is used to induce gene knockdown by inducing or promoting the degradation of the messenger RNA, also impairing the expression of the protein of interest. But what epigenome editing offers us has to do with the possibility of having a localized effect, because when we are providing a blocking antibody, this is normally done systemically. And with epigenome editing, we can do this at a specific locus without having to use any systemic approach. And also, when compared to the gene editing, with epigenome editing, we don't have to introduce any double-strand breaks in the DNA, which decreases the risk of genotoxicity and also allows or makes more favorable the multiplexing, meaning targeting more than one genes at the same time, because we know that these can cause translocations when we do so with gene editing. And another very important advantage, especially when comparing with the RNA interference technology, is that we can do epigenome editing in a hit and run approach, meaning that we don't have to provide it all the time, as it should be, for example, an shRNA. It should be integrated in the genome. Normally, it's delivered by a retrovirals construct. And with epigenome editing, we ideally don't have to do that. So I hope with this, I made clear the case of why are we interested in epigenome editing. But it would not fair if I also don't share with you some of the challenges that we are facing in this field, which are inherent to the dynamic nature of the epigenome. And one of the main challenges that we have is to achieve a long-lasting effect, meaning that we have a successful and persisting editing that stays there through cell division and also through physiological changes. For example, consider that we have successfully induced some methylation for a gene of interest that is repressing its expression. So the editing is there, but it could be that when these new cells are being produced from this cell, there is a process called replication-dependent dilution, which consists of a failure to transmit this mark, this epigenetic mark, to the new cells or new generation of cells. And with this phenomenon, we will end up losing over time the effect that we had recently induced. So this is one of the main challenges that we face, and to have more chances of achieving a successful editing that will be long-lasting, one of the approaches or one of the ideas that we had is to co-locate several epi effectors at the same time, meaning that we can have several copies of the epi effector at the same locus, or we can have different epi effectors at the same locus. And with this idea in mind, in my lab, previous PhD students developed what we call designer epigenome modifier. So I will be referring to these as them for the rest of the presentation. And this was designed with the aim of inducing gene silencing via epigenome editing. And here you can appreciate the molecular structure or molecular architecture of this molecule. And it uses a DNA binding domain that it's based on the tail platform. And at the end terminal, it is fused with a crab domain, which is, as we saw, as we previously discussed, it's a transcriptional modulator for repression. And on the C terminal, it is fused to DMT3A, which is a de novo DNA methyltransferase, and DMT3L, which enhances the catalytic activity of the DMT3A. So with this molecule, the mechanism of action would be as follows. So consider we have a basal condition where a gene of interest is being expressed under the control of a promoter that contains several CPG residues or what we call CPG islands also would be ideally. So the protein of interest is being expressed. And once we add the DEM, it binds to the entire location of the promoter, for example, and it can induce the deposition of methylation at these CPG residues and thus repress or silence the expression of this gene. So this has been published from my lab, and the application that they did in this paper was towards the silencing of two relevant genes for the HIV therapy field, for example, CXCR4 and CCR5. And they showed that the DEMs can be delivered successfully into CD4 positive cells, which are primary T cells, and it induced DNA methylation and also histone 3 lysine 9-3 methylation, which is a repressive histone mark, and it had a remarkable specificity. That's why we thought about using this technology and combining it to the CAR T cell therapy this time. And I would like to now introduce you the CAR T cell therapy concept and then explain to you why we were interested in getting into this field to make use of our epigenome editing technology. So very briefly, CAR stands for chimeric antigen receptor, and it's a recombinant protein, a recombinant receptor that it consists of several parts. So it consists of a tumor-associated antigen recognition domain, normally or most commonly in the nature of an SCFB domain. It has a hinge domain, a transmembrane domain, and it has an intracellular signaling domain that can be coupled with co-stimulatory domains. So the idea is that you would deliver this construct into the T cells, making CAR T cells, and these CAR T cells now are equipped to recognize antigens on the surface of the tumor cells and can induce the killing of these tumor cells. So the application of these in the clinics is for adoptive cell therapy, meaning that the samples, the T cells can be taken from the patient, modified most commonly by delivering the CAR in form of a retroviral particle that will carry the CAR construct and then produce and expand these CAR T cells before putting them back into the patient. This is what we call an autologous transplantation of T cells. And by now we have, at least the last time I checked, we have 5 CAR T cell products that are approved by the European organization, the EMA, and they are all aiming at the treatment of B-cell malignancies, such as leukemia, lymphomas, and myeloma. They have very high success in comparison to what was the hope before for these patients, but some of the patients do not respond and some patients relapse. And there are no CAR T cell products approved for the treatment of solid tumors. So why is this happening? It's because we still have some challenges and limitations to overcome for the CAR T cell therapy to be successful. In the case of hematological malignancies, one of the main problems is the antigen loss because of the immune pressure that is applied when the CAR T cells start killing the tumor cells that express the antigen. And another reason or another limitation or challenge is that there is normally, not normally, but in some cases, there is poor CAR T cells persistence, and this is associated with the development of exhaustion. In the case of solid tumors, it becomes even more complicated. First, there is a high tumor heterogeneity, meaning that we don't even know what to target or what to direct the CAR towards. But even if you get to find a good target, the CAR T cell still needs to traffic to the tumor site, needs to infiltrate within the solid tumor, and what it will find there is a very immunosuppressive tumor microenvironment, which also can lead to exhaustion. So with this, I think it is clear that exhaustion is a problem that is common for the treatment of both, or for the success of CAR T cell therapy for the treatment of both types of malignancies. And why is this a problem? It's because exhausted CAR T cells are in a dysfunctional state where they have decreased proliferation, decreased cytokine production, decreased cytotoxicity. And one of the most important hallmarks of this dysfunctional state is the high expression of inhibitory receptors, also known as immune checkpoints. From a very simplistic point of view, the mechanism by which this dysfunctional state is achieved, it starts with the CAR T cell recognition of the tumor cells, and in this upon this contact, the CAR T cell gets activated. When the CAR T cell is activated, it normally overexpresses these inhibitory receptors or immune checkpoints, such as LAG3, TIM3, CTLA4, PD1, and others. And upon the antigen clearance, normally the CAR T cell and also normal T cells would go back to a resting state where they do not overexpress these inhibitory receptors. But what happens is that when there is continuous antigen stimulation, these immune checkpoints remain overexpressed, and they can encounter their ligands in the very immunosuppressive tumor microenvironment. And this can drive the transcriptional reprogramming that leads to exhaustion. So that's why the aim of the project for us was to use epigenome editing to induce silencing of two major immune checkpoints in CAR T cells as a hit and run approach to render exhaustion-resistant epigenetically edited CAR T cells. So in more practical terms, what we wanted to do was to take these T cells and to treat them with the DEMS targeting these two immune checkpoints to deliver the CAR encoding virus and to obtain dual epigenetically edited, or as I will be calling them, DEPI CAR T cells. So I want to spare some words about the chosen CAR T cell model before I continue with the results. This was a second generation PSMA-directed CAR T cells that was also developed in our lab in the previous years. And this is the structure of the CAR. It contained a CD28 co-stimulation domain and a CD3 zeta activation domain, and it was delivered by using gamma retrovirus. But why this model? First of all, because PSMA is expressed at the surface of prostate cancer cell, and it has been shown to be a very good antigen to target. And prostate cancer is the second most frequent malignancy among men worldwide. And the PSMA-directed CAR T cells somehow had low persistence when tested in vivo. And not only from results from our lab, but also in other reported cases. And also, the immune checkpoint blockade therapy, which probably some of you have heard of, it seems to be unsuccessful in this case of prostate cancer, and is associated with the existence of highly immunosuppressive tumor microenvironment. So seeing all the challenges that are right now for the treatment of prostate cancer with these available technologies, this made the perfect niche for us to test a combinatorial approach. And that's why we devised this project overview, which is a very straightforward approach to obtain the dual epigenetically edited CAR T cells. So in a first stage, we attempted the single and dual editing of immune checkpoint 1 and immune checkpoint 2 in primary T cells, meaning that we took some PBMCs, we activated them, and then we electroporated them with in vitro transcribed them mRNAs. And this served us to establish the protocol and also to characterize the silencing. Then once we had this established, we moved on to attempting the dual silencing in CAR T cells. So in this case, we had to develop a protocol by which we could take CD3 positive cells, activate them, then deliver the CAR by gamma retroviral transduction, and then electroporation to achieve the DEPI CAR T cells. And with this, we established successfully the protocol, we characterized the silencing, and we also could perform some functional assays to study functional features of these CAR T cells. So I will start sharing some results with you now. This is results from attempting the single silencing in T cells of immune checkpoint 1. And what we wanted to evaluate was the long-term silencing of our T cells. So we kept the cells in culture for 39 days after the electroporation. And every 10 days, we had to perform a reactivation that served a dual purpose. The first one was to allow the detection, because the immune checkpoints, as I said before, are only expressed upon the activation of the T cells. And also, the second purpose was to challenge the stability of the silence, because every time we activate these T cells, they would receive the trigger to drive overexpression. So here I will show you results from day 19, which is the midterm of our experiments. And what we found was that at this time point, the sample that was treated with the DEM number 1, in this case, it's the name that we gave to this DEM. The sample that was treated with this DEM had much lower percentage of cells that were positive for this immune checkpoint. So this was about 70% loss of the positive cells. And we found that this was associated with also a decrease in the transcript levels of the sample that was treated with this DEM. We wanted to be sure that this was due to epigenome editing. So we took the samples and performed bisulfite sequencing of an area of about 400 base pairs that was centering the targeting position of the DEM. And here you can see a heat map of the methylation percentage. And you can see that the sample that was treated with the DEM had much higher methylation levels than the controls. Of course, we followed these cells over time, and we could see that even though the silencing was a little bit lost over time, we managed to, or we found that it was stable until this day 39 post-electroporation, meaning that the silencing was persisting despite the cell division and despite the multiple activation triggers. So we did the same for the immune checkpoint 2, and I will not bore you with those results, which look pretty much the same. And then we went on to do the dual silencing in T cells, where we again wanted to see if this was, if the multiplexing of these two immune checkpoints for the gene edit, for the epigenome editing was stable also over time. Here you can see also data from day 19 post-electroporation, and the sample that received the dual treatment had lower levels of expression or lower levels of cells that were positive for immune checkpoint 1 and immune checkpoint 2. This was also accompanied by a decrease in the transcript levels. And we also were interested to see what happens to the immune checkpoint 1 when we target immune checkpoint 2 and vice versa. And here you can see that indeed when we provide the DEM number 1, we only see a decrease in immune checkpoint 1. And of course, this is the dual silencing. So here we also see it, but not when it's not provided. So meaning that our silencing was specific to immune checkpoint 1. And the same we found when we checked the expression levels of immune checkpoint 2. And so it was only decreased when we provided the DEM number 2. In terms of long-term silencing, we again followed them until day 39 and found that this silencing was pretty much stable for the single treatment and also for the that I already showed before, but here it's data from the dual treatment. So having established the dual silencing in T cells, we moved on to the generation of the DEPI CAR T cells, which allowed us to perform an in vitro characterization using a target positive cell line. And we performed two types of experiments, one that we call acute exposure and another one called chronic exposure. So in the acute exposure, we co-cultured the DEPI CAR T cell with the target positive cells for about 24 or 48 hours. And in the chronic exposure, we performed a co-culture of 12 days, meaning that we had 4 rounds of antigen encounter. And with the purpose of studying how stable was the epigenome editing in these CAR T cells. So we characterized the functional features of the DEPI CAR T cells to know if our treatment was causing any impairment to the functional activity of the CAR T cells. And one of the main properties that a CAR T cell should have is the ability to kill the target cells. And here you can see the results from our cytotoxicity experiment when we use a target positive cell line and a target negative cell line. And you can see that there is not much difference between the samples that were epigenetically edited, are either single with DEM1 or DEM2 or dual in comparison to the parental CAR T cells. We found no unspecific phenomenon on the target negative cells. We evaluated also the differentiation of these CAR T cells toward the effector and the effector memory phenotype upon the antigen encounter and found no differences among the different sample groups. And we also measured some cytokines that are released upon the CAR T cell activation and found again not much differences among the different sample groups, meaning that our epigenetically edited CAR T cells could perform as good as the parental CAR T cells that were non-modified. Then in terms of the long-term silencing of these on these DEPI CAR T cells, as I said before, we had this chronic exposure experiment, and every third day we had to add more target cells because the CAR T cells were clearing them out. So in this case, we had to co-culture for a total of 12 days, which was the endpoint of the experiment. And at this time point, we collected the cells and wanted to see if the silencing was still there. So here I'm showing you the relative mRNA levels of the immune checkpoint 1. And you can see that in both either the single or dual targeting, we did see that it was still decreased at this time point. And moreover, the bisulfite sequencing at this time point also showed us that the methylation was highly increased in the samples that received the DEMs, either single or dual for this immune checkpoint 1. And something very similar happened for the immune checkpoint 2, where we also saw a decrease still at the transcript level, and this was also associated or accompanied by an increased methylation in this single or dual targeting sample. So in summary, we can say that epigenome editing is a novel technology that allows precise modulation of gene expressions. And this was a unique report of multiplex epigenome editing targeting 2 very highly dynamic genes in primary T cells. This was totally compatible with the CAR T cell technology in a hit and run approach, and it's also applicable to other CAR T cell models. And the induced silencing persists through replication-dependent dilution and physiological challenges, such as it was the repetitive activation that we perform, and does not impair the functionality of the CAR T cells in vitro. And with this, I would like to thank you for your attention, and I will be happy to take your questions.
Thank you so much, Maria. This was a beautiful talk, very clear, very visual. I think the audience really has the main essence and the main message because it was very clearly explained. Thank you.
Thank you.
So first of all, because then we're going to stop sharing, I would like to announce that we have on the 21st of April another webinar, which will be given by Marine Laurent from Geneton, and she's going to talk about ex vivo editing of hematopoietic stem cells for erythroid expression of beta globin changes and therapeutic proteins. So I think we can now go to the questions. So you can stop sharing, Maria. Thank you. I really found this an amazing technology, because one of the things that you clearly pointed out in the beginning is that double-strand breaks can lead to toxicity. We know more and more that the CRISPR-Cas9 system is a very beautiful gene editing tool, but it also has backdraws, and that's something which is clear. They can be a little bit of targets. They can also be even chromosomal rearrangement. So I think this is a really a very nice other tool to modify, especially since the new gene editing tools like prime editing and base editing are much less efficient for the moment, at least in primary cells. So it's not so easy to do this. One of the questions that I had is, could you, if you see by any chance that there is something not cool about introducing an epigenetic mark, can you erase it again?
Yeah. So this is one of the, this is something that can be a disadvantage in the sense when you want to add it, because it can be removed. So it is reversible because there is no change on the DNA sequence. So it could be theoretically reverted, and we have done this also in the lab. We have silenced some cells, and then we had added the reverse treatment, let's say, and this works. But I mean, when we talk about clinical applications, of course, these might be more challenging because then we would also have to do some targeted epigenome editing, or there are also available what are called epi drugs that can overall change the epigenetic landscape and are actually being used in the clinics. So theoretically, let's say, yes, it is possible.
Then I have a specific question, because you here use in a previous paper and also for the modification of the CAR T cells, the DNMT3A. Why is this particularly attractive?
Yeah, it is particularly attractive because so far, as I said, we don't yet speak very fluent epigenetic regulation as a language. And so we don't yet know exactly how are these epigenetic marks passed from one cell to another, from one individual to the next generation of individuals. So this is very complicated. But what we do know is that it seems that DNA methylation is the one mechanism that is more likely to be passed on to the next generation of cells. So that's why this DNMT3A is so attractive for epigenome editing, because it allows us to introduce de novo methylation. And I would say this is the reason why us and many other groups are working with this enzyme. I cannot hear you now, Els. You are muted.
Thank you. I had another question because I was a bit surprised. If I understood it well, all this work was done in human T cells, right? But nevertheless, you use a gamma retrovirus. Is this a bit historical because of the lab where you are in, or is there a particular reason for that?
So let's say we can provide the CAR construct in different ways. For example, now they are identifying, say, harbors in the genome where you can integrate the CAR. Also, some are doing it in the track locus and so on. But since this was work done in our lab using gamma retrovirus, it's let's say historically the way to go to provide the CAR construct. So far, we decided to use this, let's say, more easy strategy, which we know the CAR is then inserted somewhere in the genome where we have no control of. But I think it works good enough for our purpose of characterizing the effect of the epigenome editing. But yeah, nevertheless, in the future, this might move towards a different direction, having more control over the CAR expression.
Yeah, maybe inserting it into a specific locus or and or moving to lentis. But in the end, we know that gamma retroviral vectors have caused problems in stem cells, in hematopoietic stem cells, but in T cells, there is no real proof for that. So I don't know if it's that problematic for the moment, right? So I saw that you are targeting the PSMA epitope with the CAR. Is in this case, I'm talking about ex vivo, not in vivo, but is the CAR T cell already a powerful CAR in this context? Is it really a strong CAR T cell effect that you have?
Yes, so in vitro, we do see complete clearance of the target cells that are expressing the PSMA antigen. And in vivo, as I mentioned before, there is still some challenge to overcome, in the sense that the CAR T cells seem to get exhausted. So it seems that it's probably not related to the antigen itself, but more related to the tumor microenvironment and the fact that it's very immunosuppressive. So I think it is a very good target. And I have hopes that at some point we will have a CAR T cell product targeting PSMA, because it is really highly expressed in prostate cancer cells. It can also be expressed at very low levels in other tissues, but so far it seems like it's one of the best candidates to treat prostate cancer.
Okay. And one thing I was wondering is, I mean, I guess that here you also transduced your CAR T cells and expand them in a protocol where you stimulate 328. So
Yes.
where you have high expansion. Don't you think that, I mean, people are working a lot on this issue, that it might be better to do this with a milder kind of stimulation? Are you doing things in that direction?
So we do know that we have to be very careful with the way we treat our cells in culture when we want to produce a CAR T cell product. Of course, we know that we can use certain cytokines like IL-15 and IL-7 to preserve their central memory phenotype. And this first activation step, that we need to get the cells in a state where we can transduce them and electroporate them. I think this can be probably optimized, but so far it's the only protocol that allows us to see the treatment or to induce either the gene knockouts or the epigenome editing. So far, I think we need to spend some more time, but I think in the future we might come up with new delivery methods that can allow us to preserve the naive T cells before going into the patient. But yeah, I think everybody or most of the people are probably doing the same. We do need this first activation.
Yeah. For the moment, it's still the products that are now available.
Because also the T cells don't expand when they are not activated. So it's, but yeah,
no, it's true.
It's tricky.
It's a dual problem that, or a question of, do you want to maintain them long in the patient? But if you want to make them act soon and rapid enough in cancer patients that don't have a lot of time for the treatment, I agree that it's not easy to answer this question. And then I have a question which is really that I had a clarification for myself. The dual CAR T cells, so the Epi-CAR T cells, are you able to purify them? No. You are talking about a mixture, right?
Exactly. Throughout the experiments, I had a mixture of cell population. Like some of them were only silenced in one, probably some only silenced in the second one, or silenced. Hopefully, in some events they are silenced in both. I mean, I do see this when I check the dual positive cell percentage, which is also decreased when we do the dual treatment, but. So far, we have not spent so much time thinking about how to enrich this population, because we also know that these immune checkpoints are important. And we believe that having these kind of 50% silencing can allow us maybe to be at the sweet spot where we don't have a complete abrogation of these immune checkpoints, but somehow still preserve some of them. And we are very interested to see how would these perform in vivo, actually. So we are now trying to move forward in the next step, and let's see, maybe we could compare it with a knockout where we can have a very high abrogation, and then see if it's what really works better.
Okay, yeah,
it's very interesting.
Yeah, because another way to go is really to knock out the gene completely.
Yeah,
but I don't know if that is so secure either sometimes.
Yeah, some authors are reporting that it might not be. I mean, there are some papers saying that if you abrogate completely PD-1 expression, then the T cells cannot proliferate so good anymore, and so on. So there are still open questions about this.
Okay, but I think, yeah, that you're right. Probably in vivo, on the long term, you might have a bit of enrichment of those, or you might have enriched or only have left those CAR T cells that have the...
That would be very, very interesting to know.
I agree with you. So Maria, I thank you very much also for your answers to the questions. And again, I really want to congratulate you. And I think that the audience agrees with me that this was a very instructive webinar and presentation. Thank you so much.
Thank you very much for the invitation and for the feedback.
Bye-bye.
Bye.
And I hope to see you all soon again for the next one.